Okay, ladies and gentlemen, today we're going to talk about Mendelian genetics, or something that we like to refer to as the patterns of inheritance. So the first thing I'd like you to do is to think about who it is that you look like. Every one of us has been told that perhaps we look like our mother, or perhaps we look like our father, or maybe you look like a brother or a sister. But what I want you to think about is when people tell you you look like them, is it that you look exactly like them? And have you ever wondered how it is that this kind of thing happens? You see, this is an important concept in the overall scheme of biology, but it's not something that we necessarily think about. We just sort of accept that we look like our parents, or we accept that we look like our family members. And the first step to understanding how that happens is to talk about what we call the patterns of inheritance. The patterns of inheritance start with a scientist in the late 1800s by the name of Gregor Mendel. Mendel was a monk, spent a lot of time in the garden, and he also was a mathematician. To be exact, he was a statistician. He was very into statistics, probabilities, the likelihood that things would happen. And since he was a gardener, he actually happened to wonder, how is it that plants could be bred to have certain traits? So what he did, like any good scientist, is he used his knowledge, which in this case was mathematics and statistics, to collect evidence so that he could draw a conclusion about what it is that was actually going on. So the first thing that Mendel did is he started by taking a look at the plants he wanted to work with. Now he chose peas. Peas were a good choice. They have a lot of different traits, and their traits tend to be either one thing or another, which by happenstance actually turned out to be a very important thing for his experiments. You'll notice here on this table, you can see here's a trait, flower color. And what you see over here, there are two options for flower color, purple or white. Let's take another one. For instance, seed color, which would be the pea itself. And seeds, or peas, can be either yellow or green. And you'll notice if you look up and down this list that all of the traits are one thing or another. And so what we call these traits are phenotypes. And a phenotype is defined as the expression of a trait. For our purposes, it's what it looks like. So the flower color looks purple, or it looks white. Purple is a phenotype. White is a phenotype. The stem is tall. The stem is short. Tall is a phenotype. Short is a phenotype. So the next thing that Mendel did is he took these peas and he actually crossed them. And we use this term crossed here to mean that he bred them with each other. So what he tended to do was he would actually cover the peas in plastic bags so their pollen wouldn't spread all over. And he would actually use paintbrushes to take the pollen from one plant and use it to fertilize another. And what this did is he would take plants that had one trait and plants that had another trait, breed them together. And he ended up with all plants that had the same trait. In this case, he took a tall and a dwarf plant, and all of the offspring were were tall rather. So then what he did was he took that generation right here and he bred them with themselves. That's why it says selfing right here. And the next generation had tall, and short plants, but the tall plants were far more common. So what this meant was that the expectation that Mendel came into this with was incorrect. Because at the time, scientists and Mendel himself assumed that when you took two traits, like tall and short, and bred them together, you would end up with something in the middle, a medium. And you could see from this pattern shown here, that's not what happened at all. So what Mendel wanted to do, like any good scientist would, is to determine what it is that actually happened. So Mendel collected thousands of pea plants, and he repeated his experiments numerous times. And he would do the experiments with the flower colors, then he would do the experiments with the plant height, then he would do the experiments with the seed color or the seed shape. He would do it for each of the traits. And after he collected all of this data, he came up with an interesting hypothesis as to what it was that was actually going on. He thought that sexually reproducing organisms actually have two copies of each trait, and he called those copies alleles. You can see that term right here. Okay. Each parent gives one copy of their trait to the offspring. So each parent actually gives one allele to their children. And so then the children have two alleles, one from each parent. He also realized that each trait has a dominant allele that can hide the other allele. And we call the other allele recessive. We're going to explore that a little bit as we move forward. So what we see over here is Mendel's cross. So what Mendel did is he would take two parents that were, and the term we use is true breeding. So this parent right here has two copies of the tall allele. And this parent right here has two copies of the short allele. And we call this the P generation, because the P generation are the parents. And this tall parent would give one tall allele to the offspring, because that's all it has to give. This short parent would give one short allele to the offspring, because that's all it has to give. And when these two alleles come together, this offspring has one of each allele, but is still tall. And so that's what dominance is, when one allele, in this case the tall, can hide the other, in this case the short. And this first cross is called the F1 generation, which stands for the first filial or offspring. So Mendel then wanted to see what would happen if he mixed this plant with plants that are similar. So he crossed these two. And what you see down here is a box. I'm sure you've seen it before. It's called a Punnett square. And in this Punnett square, you see all the possible ways that this big T and this little t, see, big T, little t, from a mother, and big T, little t, from a father, could combine. Big T and big T could join together. Big T and little t could join together. Big T and little t could join together, or little t and little t could join together. And this is what we call a Mendelian cross. Now, what happens here in a monohybrid cross, mono meaning one and hybrid meaning mixture, so we're actually mixing the crosses of the traits of two organisms, is you take the gametes, which you can look down here at the bottom, which are the individual alleles given by the parents, and you separate them. One gamete, or rather both gametes from one parent on the side, and both gametes from one parent on the top. And then this four-square box in the middle, you take one gamete from the parent, one gamete from the parent, and combine them. One gamete from this parent, one gamete from this parent, and combine them. This gamete, and combine them. This gamete, and combine them. And what this does is these four boxes give you all the possible ways that the gametes can combine. Here you have one big A and one big A. In this case, big A is normal skin tone. And so since this person only has the normal skin tone allele, they have normal skin tone. Over here you have one big A, one little a. That's normal skin tone and albino skin tone. This person has one normal, one albino. Since albino, or rather normal, is dominant, this person looks normal. This person has one normal, one albino. Notice that the order does not matter. And this person, since they have one normal, they appear normal. Now over here in the bottom corner, you'll notice that this person has one albino and one albino. So this person actually appears albino. And so the phenotypes here are normal, 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 albino. And so what we've learned is out of 4, 3 of them are normal. You can see that right here with the 3. And 1 of them are albino. So that's what we call a phenotype ratio. For every 3 normal people, you will have 1 albino people. Now, it doesn't work that way, but this represents the probability. Now down here, you're seeing something different called a genotype. And the genotype actually represents the different allele combinations. And we are representing the alleles here with letters. And so there is one possible combination that's big A, big A. There are 2 possible combinations that are big A, little a, big A, little a. And there is one combination that is the albino, little a, little a. And you can see down here you have gametes, the individual alleles given by the parents, the phenotype ratio, which is the relationship between the different possible appearances of the trait, and the genotype ratio, the relationship between the possible allele combinations. Now over here you see a Mendel cross, and what we have down here are simply our definitions from before. P1 being the parental generation, you can see that up here. F1 being the first filial generation, or the offspring when you cross the P1. F2 is the second filial generation, which is down here, and that's the offspring when you cross the F1 with itself. Now, there are two key terms here. A heterozygous individual is an offspring who has two different alleles. So if you were to go back to our previous one, which is right here, big A, little a, big A, little a, that would be a heterozygous individual. Going back here, another key vocab term we have here is homozygous, or purebred, and that is offspring who have identical alleles. So again, if we go back to our example over here, little a is homozygous, and big A is homozygous. Now, after doing all of this research and learning all of these things from viewing the phenotypes of these plants, Mendel came up with some ideas about what was happening. The first one we call the law of segregation, which means that the alleles of the parent actually separate randomly before the parent reproduces. That's why they only give one to their offspring. The second thing is known as the law of independent assortment, which means that the parental alleles combine randomly and they don't influence each other. So the fact that a dominant allele, that a parent has two dominants, doesn't matter when they separate. The fact that a parent has one dominant and one recessive doesn't affect how they separate. And it's our understanding and what Mendel has given us with his experiments of these laws that allow us to set up Punnett squares to determine all probable outcomes with a pretty high level of confidence. Speaking of probable outcomes, one of the things that I want to discuss is this idea of probability. All right, it's very important for you to understand that when we discuss Mendelian genetics, we are not telling you what will happen. What we're actually telling you is how likely something is to happen. Okay? When we fill out a Punnett square, we're actually giving the probability of each possible result. So what happens is when we express that probability, we express that probability as either a ratio, which is written as two numbers on either side of a colon, sometimes three numbers. It can be expressed as a fraction, something like 1/2, or it can be expressed as a percent, something like 50%. And in fact, the ratio, fraction, and percent that you see here are all equivalent. So a ratio of 1:1 is the same as saying 1/2, which is the same as saying 50%. I understand this can seem a little mysterious right now, and we'll actually do a number of problems and exploring of this in class. Over here, we have another key idea that's important for this. Mendel's ideas work because some genes are dominant. So every trait has two alleles, or two versions of itself. Pea plant height, for example, is the trait. Tall is one allele, short is another. Pea flower color is the trait. White is one allele, short is another. Dominance is when one trait phenotype can hide the other phenotype. So that's why a pea plant could have a tall allele and a short allele, but still appear tall. Dominant phenotypes are expressed when one or more alleles is present, so one or two. And recessive phenotypes are only ever expressed when both copies are of that allele. So in essence, what you're going to find out here is that Mendel was able to use his understanding of math to breed pea plants in large numbers and look at their phenotypes. And what he realized is that all organisms that reproduce sexually have two copies or two versions of each trait, and these versions separate and one is given to the offspring. And when you mix those one alleles from each parent, you end up with variety.